You have studied what are the various explosives, the types and how to manage the crime scene. But when you take these evidence of bombs and other explosive material into the forensic lab, then how to deal with the inorganic anions and cations which are there as components of these material or the evidence that you have collected. So, this module shall focus on the analysis of inorganic anions and cations of the post-blast residues. In this module, we shall try to understand how the various chemical tests are done for the inorganic anions by making their water extract for the anions like chloride, sulphate, nitrite, thiocyanate, chlorate, thiosulphate, perchlorate, sulphite and phosphate. And similarly, we will try to understand what are the chemical tests for cations making their water extract again and testing for the potassium, barium, strontium, aluminium and the antimony cations, because these are the ones which are commonly found at the scenes where explosion has happened. And we will also see how the flame test for sodium, potassium, barium, calcium and strontium can be done. In addition, we will do some chemical tests for inorganic anions making their pyridine extract, and there comes the sulfur, and then we will also see how the TLC of cations can be done. We will also see how the chemical test for some anions in pyridine extract can be done like for sulfur, and how the thin layer chromatography of some cations like aluminium, zinc, arsenic, barium, mercury and lead can be done. So, you will be able to comprehend the various chemical tests for detecting these cations and anions in a post-blast residue. Let us begin with the analysis of post-blast residue. Tests for explosives are carried out to detect and identify the ions present in the explosive material. The test must be carried out along with the reference standard as blank. So, when you are in a forensic lab, you take a known cation, carry out that test, and the evidence or the sample you have collected from the scene, also you conduct the similar test and just compare that. You will be guiding you whether the test is positive or not. And let us see what are the various tests that you have to perform in the forensic lab. So, dear students, with this background, let us try to understand how the various tests you can perform in a forensic lab. And it is a very simple chemistry. You have the anions, you make the corresponding reagents added, there will be color formation or some precipitate, something which will be very indicative will be observed, that you have to observe and just compare with your sample that you have got from a reference one, and you will be able to detect this. So, let us try to understand these tests in more detail with the help of graphic and visuals.
Chemical tests for inorganic anions, water extract. First is tests for chloride. The following three reagents are required for detecting chloride ion in an explosive. Reagent A, it is made by dissolving 128 milliliters of concentrated nitric acid in 500 milliliters of distilled water and then diluting it to 1 liter. Reagent B, it is prepared by dissolving 16.99 grams of silver nitrate in 500 milliliters of distilled water and diluting it to 1 liter. Reagent C, reagent C is prepared by dissolving 95 grams of lead acetate trihydrate in a mixture of 500 milliliters distilled water and 10 milliliters glacial acetic acid, and then diluting the solution with water to 1 liter. Take 1 ml of water extract in a clean test tube. Put 2 to 3 drops of reagent A, followed by the addition of 2 to 3 drops of B reagent. The presence of chloride ions is shown by the formation of white precipitate. This white precipitate is soluble in ammonium hydroxide and insoluble in nitric acid. Another method to test for if chloride ion is present or not is carried out by taking 1 milliliter of extract in a fresh test tube, followed by the addition of 3 drops of C reagent to it. The presence of chloride ions are indicated by the formation of white precipitate. Next is test for sulfate. Before performing the test for sulfate ion, we have to prepare the following two reagents. Reagent A. It is prepared by dissolving 61.1 grams of barium chloride dihydrate in 500 ml of distilled water and then diluting it to 1 liter. Reagent B. It is freshly prepared by dissolving 0.5 grams of sodium rhodizonate in distilled water. The test is carried out by taking 1 ml of extract in a clean test tube and then adding 2 to 3 drops of dilute hydrochloric acid to it. The test tube is then placed in a water bath at 100 degrees Celsius for 5 minutes and 2 to 3 drops of reagent A is added to it. Formation of white crystalline precipitate indicates the presence of sulfate ion. Tests for nitrite. Before analyzing the presence of nitrite in post-blast residue, the following four reagents are to be prepared. Reagent A. Sulfur-anilic acid 1 gram is dissolved in 100 milliliters of warm 30% acetic acid. Reagent B. Take 0.3 grams of alpha-naphthylamine and dissolve it in 70 milliliters of distilled water, and it is then kept for boiling and is filtered. Add 30 milliliters of glacial acetic acid to it. Reagent C or indole reagent. To prepare this reagent, 0.015 grams of indole is dissolved in 96% alcohol, 100 ml. Reagent D. It is prepared by slowly adding 500 milliliters of water under constant strain to 445 milliliters of concentrated sulfuric acid. When the mixture cools down, again dilute it with water to 1 liter. The test is carried out by taking 1 drop of neutral or acid extract on a clean spot plate. This step is followed by the addition of reagent A and a drop of B reagent. The presence of nitrite ions are indicated by the formation of red color. Another method for testing the presence of nitrite ion is done by taking 1 drop of the extract in a semi-micro test tube and then add 10 drops of reagent C, followed by adding 2 drops of reagent D to it. A purplish red color indicates the presence of nitrite ion. Tests for thiocyanate. To test the presence of thiocyanate in the given sample, firstly, we have to make the following 2 reagents. Reagent A. It is prepared by dissolving 146 grams of cobalt nitrate in 500 milliliters of distilled water and then diluting it to 1 liter. Reagent B. It is prepared by dissolving 62.42 grams of copper sulfate in 500 ml of distilled water and then diluting it to 1 liter. Test for thiocyanate is carried out by mixing 1 drop of the test solution in a microcrucible with a very small drop of reagent A and then evaporating it to dryness. The residue will be of violet color if thiocyanate is present and the color fades away slowly. Blue or green color is obtained when drops of acetone is added to it. Another method for testing the presence of thiocyanate is carried out by taking a few drops of pyridine in a clean test tube. To it, 3 to 4 drops of reagent B is added, followed by 2 ml of chloroform and 5 to 6 drops of the extract. Shake the mixture vigorously. The chloroform layer will acquire a green color, which indicates the presence of thiocyanate. Test for chlorate. To confirm the presence of chlorate in the given sample, we will have to prepare the following 3 reagents. Reagent A. It is prepared by dissolving 1 gram of aniline sulfate in 100 milliliters of distilled water. Reagent B. Take equal volumes of saturated manganese sulfate solution and syrupy phosphoric acid and then mix them properly. The solution so formed is labeled as reagent B. Reagent C. 1% alcoholic diphenyl carbazide solution is used as a reagent C. Test is carried out by taking 1 ml of extract on a spot plate and is left for some time. Cool it and add concentrated sulfuric acid 2 to 3 drops, followed by 2 to 3 drops of reagent A. The presence of chlorate ions are indicated by the formation of blue color. Second procedure for testing for the presence of chlorate ions are carried out by taking 1 drop of the extract in a microcrucible and added to it is 1 drop of the reagent B. Briskly warm the solution over a microburner and allow it to cool for some time. Appearance or violet coloration can be seen. Very pale coloration may be intensified by adding a drop of reagent C. A deep violet color is formed due to the oxidation of diphenyl carbazide. Test for thiosulfate. 2 reagents are prepared to test the presence of thiosulfate. Reagent A. It is prepared by dissolving 2 grams of mercury chloride in 100 milliliters of distilled water. Reagent B. Reagent B is prepared by treating a little nickel nitrate solution with ethylene diamine until a violet color appears. This violet color appears due to the formation of the complex nickel ethylene triamine ion. Take 1 drop of the water extract in a clean test tube. Add 2 drops of reagent A and mix it thoroughly. Then add little amount of potassium chloride to it. Take a blue litmus paper and dip it in the solution. The presence of thiosulphate can be indicated if the blue litmus paper changes to red in color. Put 1 ml of the neutral or slightly alkaline water extract in a fresh test tube. After this, add 2 to 3 drops of the reagent B to it. Formation of crystalline violet precipitate indicates the presence of thiosulfate. Next is tests for perchlorate. Reagent A. It is prepared by dissolving 28.8 grams of zinc sulfate heptahydrate in 100 ml of distilled water. Reagent B. It is prepared by dissolving 10.1 grams of potassium nitrate in 100 milliliters of distilled water. Reagent C. Reagent C is prepared by dissolving methylene blue 0.05 grams in 100 ml of distilled water. To detect the presence of perchlorate in an explosive, take 1 drop of water extract of post-blast residue on a piece of a filter paper. Impregnate it with reagent A and reagent B, and then spray reagent C on it. Violet spot appears, which indicates the presence of perchlorate. Test for sulphite. Reagent A. Saturated solution of zinc sulfate is used as reagent A. Reagent B. It is prepared by dissolving 10.4 grams of potassium ferrocyanide in 100 milliliters of distilled water. Reagent C. It is prepared by dissolving 1 gram of sodium nitroprusside in 100 ml of distilled water. The test is carried out by adding a drop of reagent B to a drop of reagent A, followed by a drop of reagent C. White zinc ferrocyanide precipitates is formed, and a drop of neutral water extract is added to it. Red precipitate appears, which confirms the presence of sulphite in it. Test for phosphate. Reagent A. It is prepared by dissolving 0.5 grams of ammonium molybdate in 10 milliliters of distilled water, and then 3 milliliters of concentrated nitric acid is added to it. Reagent B. It is prepared by dissolving 6.5 grams of benzidine in 10 ml of glacial acetic acid and diluted it to 35 milliliters with distilled water. Reagent C. Dissolve 16 grams of sodium hydroxide in 100 milliliters of distilled water, and the solution so formed is used as reagent C. To identify the presence of phosphate, put a drop of water extract on a spot plate. Add a drop of A reagent to it. Wait for 30 seconds, followed by the addition of 1 drop of reagent B. Wait for half a minute. Now add saturated solution of sodium acetate, 3 drops, to it. Blue-gray color indicates the presence of phosphate. Now we will study about the chemical tests for cations. First is test for potassium ion. Reagent A. It is prepared by dissolving 0.2 grams of dipicrylamine in 2 milliliters of 2 normal sodium carbonate and 15 milliliters of distilled water. Filter paper strips, which are placed in reagent A, should be freshly prepared and then dried. Reagent B. It is prepared by dissolving 6.73 grams of sodium cobalt nitrite in 100 milliliters of distilled water. Place 1 drop of water extract on paper of reagent A and then add 0.1 normal nitric acid, 2 drops. A red stain at the side of the spot appears, while all other parts of the reagent paper turn bright yellow, which indicates the presence of potassium ion. Second method for confirming the presence of potassium is done by taking 5 drops of water extract in a watch glass and then evaporate it to half of its volume and then leave the remaining residue to cool down for some time. Then add 4 drops of reagent B and warm if necessary. Yellow precipitate is formed, which is insoluble in dilute acetic acid. This indicates the presence of potassium ion. Test for barium ion. Reagent A. It is prepared by dissolving sodium rhodizonate 0.2 grams in distilled water, that is 100 ml. Reagent B. It is prepared by diluting 4.5 ml of concentrated hydrochloric acid with 100 milliliters of distilled water. Put a drop of neutral or slightly acidic water extract on the filter paper, followed by the addition of reagent A. The presence of barium ion is indicated by the presence of reddish-brown spots. A brilliant red acid salt is formed on addition of B reagent. In case barium is absent, this red color will disappear. Next is test for strontium ion. Reagent A. It is prepared by dissolving 0.2 grams of sodium rhodizonate in 100 milliliters of distilled water. Reagent B. It is potassium chromate saturated solution. Put 1 drop of neutral extract of sample on a filter paper or on a spot plate. Add a drop of reagent A. A brownish red coloration or precipitate is formed, which indicates the presence of strontium. If barium is also present, then impregnate the filter paper with reagent B and dry it. Place a drop of extract on this paper and after a minute, place a drop of reagent A on the moistened spot. Brownish red spot or ring is formed, which indicates the presence of strontium ion. Flame test for sodium, potassium, barium, calcium, and strontium. Put drops of the water extract in a beaker till it evaporates and is dry. After this step, cool it. Take concentrated HCl in a watch glass. Perform flame test with the help of platinum wire in the non-luminous flame of the burner. The response of various ions to the flame test is given. Next is test for aluminium. Reagent A or aluminium reagent. Reagent A is prepared by adding saturated solution of morine in 5 milliliters of methyl alcohol in a small bottle. Reagent B, quinalizarin reagents. Dissolve about 0.25 grams of aluminum in distilled water, that is 250 ml. Reagent C, 1258 tetrahydroxy anthraquinone is used as reagent C. To prepare reagent paper, immerse quantitative filter paper in the solution of quinalizarin, which is attained by mixing 0.01 grams of quinalizarin in pyridine, 200 ml. Now dilute it with 18 milliliters of acetone. Take water extract and add a small amount of 1 normal acetic acid in order to make it slightly acidic. Place 3 drops of the acidified solution in a black spot plate. Now add 2 drops of reagent A. Observe the spot under UV light. If green fluorescence appears, it indicates the presence of aluminium. Next are the chemical tests using alkali extract. Alkali extract is used for analyzing the presence of antimony and sulfide ions. Test for antimony. The method of preparing the reagent is to dissolve 0.01 grams of rhodamine B in 100 milliliters of distilled water. Take a small portion of sodium hydroxide extract in a spot plate. Add 1 to 2 drops of concentrated hydrochloric acid and a few milligrams of sodium nitrate. Now put 10 drops of the prepared reagent and mix it. A change of color from bright red to violet indicates the presence of antimony. Test for sulfide ions. Reagent A. It is prepared by dissolving 0.1 grams of methylene blue in 100 milliliters of distilled water. Reagent B. It is prepared by dissolving 0.1 grams of sodium nitroprusside dihydrate in 10 milliliters of distilled water. Use freshly prepared solution before carrying out the test. Reagent C. It is prepared by dissolving 135 grams of ferric chloride hexahydrate in 500 milliliters of water. Put few drops of concentrated hydrochloric acid and dilute with water to make it up to 1 liter. In case the solution turns dark, add more hydrochloric acid. To conduct the test, place a drop of alkali extract on a spot plate. Add a drop of concentrated hydrochloric acid and mix it properly. Add 2 to 3 drops of reagent A to it, followed by a drop of reagent C. A clear blue coloration appears after a short time. Mix a drop of B reagent with the alkaline extract on the spot plate. The presence of sulfide ions is indicated by the presence of violet color. Next, we will study about the chemical tests for inorganic anions, pyridine extract. First, we have test for sulfur. To test for the sulfur, take a small portion of pyridine extract in a small test tube and then boil it for 1 minute. Add a drop of 2 molar sodium hydroxide to it. A blue to green color for low and red to brown for high concentrations of sulfur is observed. Next is thin layer chromatography of cations. Thin layer chromatography of cations such as aluminium, zinc, arsenic, barium, mercury, and lead compounds is carried out by using N-butanol and glacial acetic acid in 8:2 ratio as a solvent system. This is used as a solvent system and samples are applied on the pre-coated TLC plate of size 20 centimeters into 20 centimeters into 0.25 millimeter silica 60F. Spraying reagents dimethylamminobenzaldehyde and pyridine. A. Dimethylamminobenzaldehyde is prepared by dissolving 5 grams of dimethylamminobenzaldehyde and 5 grams of ammonium thiocyanate in 50 milliliters of methanol. B. Pyridine reagent is prepared by adding 2 milliliters pyridine and 5 grams of ammonium thiocyanate in 50 milliliters of methanol. The table shows the response of various cations to the solvent system. N-butanol is to glacial acetic acid in the ratio of 8:2, and after spraying the TLC plate with dimethylamminobenzaldehyde reagent and pyridine reagent.
So dear students, let us now summarize what we have learned in this module. You have learned that the test for explosive can be carried out very easily by identifying the ions present in them, which may be cation or anion. The test must be carried out along with the reference standard as the blank. This is always you should keep in mind. And we have understood what are the various reagents and the procedure for carrying out the chemical tests for ions such as chloride, sulphate, nitrite, thiocyanate, chlorate, thiosulphate, sulphite, phosphate as anions, and also if we look at the cations that we have gone through to understand the tests how they have to be performed, they are the potassium, barium, strontium, aluminium, antimony, sulphide, etc. And all these tests, majority of them are carried out in aqueous solution, and some pyridine extract or alkali extracts are also made. In addition, we also studied how the flame test for sodium, potassium, calcium, barium, strontium will give corresponding color in flame, that is you will get golden yellow for sodium, violet for potassium, crimson for strontium, and so on for detecting these cations. So, after knowing all these tests, I am sure you will be able to analyze the post blast residue and find out what are the cations and anions present and correlate with the cations and anions which are present otherwise in that type of explosive material. It will give a clue, so that is what was the type of bomb or the explosion material that was responsible for creating that explosion in the scene of crime.